So my name is Ellen. I'm a postdoctoral fellow at Colorado State University, hoping to implement evolutionary ecology theory into biological control. And Amanda Stahlke is a first-year grad student at the University of Idaho, and she is studying bioinformatics. And together we hope to create a very full picture of what is happening with the Tamarisk leaf beetle as it is introduced and making its way across the southwestern United States. So first, classical biological control involves the release of natural enemies to control an invasive pest. We all know that it's a very effective long-term method with very high cost-to-benefit ratio and minimal use of resources. As a very brief example, tansy ragwort in the northwestern United States, an invasive species. They brought over the cinnabar moth, and the USDA now expects over 80% control in new infestations. So with very little money, we can have a long-term control program. The problem, a big problem with biological control programs is the success rates. And so we need to start thinking about ways to improve the success of different programs as they get off the ground. So one thing that we think about a lot is evolution, and evolution happens. As these agents are released into the field, they will undergo many different evolutionary processes, such as different levels of gene flow, different levels of genetic variation depending on where they came from and the population sizes as they're introduced, and also, of course, natural selection in their new environment. Another evolutionary process that they might experience is hybridization. Now you might think, well, normally we release one species, and so hybridization, what does that have to do with anything? But there's two things I would say. One, as you may or may not know, and if you didn't, I'm sorry to ruin the surprise, but there are 4 species of salt cedar beetle that were released, and so we could expect maybe there's hybridization there. But even if you don't release different species, often they release different ecotypes, which are the same species, but they come from very different geographic areas. And as they mate with each other, genetic variation can increase, and similar effects that are seen with hybridization can occur. So it's definitely something we need to think about. So Amanda is going to give a whole second part of the presentation where she's going to discuss how we can quantify the hybridization using really advanced genetic techniques. But I'm going to be talking about lab experiments that I am doing right now at Colorado State University. And our main question is, with this particular system, how do hybrids perform compared to their parents? We're going to be looking at different life history traits of the hybrids versus their parents. Also importantly for biological control, the potential for host range expansion, which is simply the addition of a new host to an already existing diet. And also, I will talk a little bit about male-female directionality, because when you have hybrids, you can have different genetic contributions from the male or female of one species, and it might create different patterns. So I just want to really stress that this is very important for biological control. We can have at least 3 different outcomes, right, where hybrids have higher fitness, lower fitness, or the same fitness as their parents. But more importantly, you can also have interactions between different crosses and the environment. So where you might see, and I'm so glad that she just gave that talk before, because it just occurred to me that sometimes you have establishment of populations or not, and you don't really know why. And looking at this kind of evolutionary process with different levels of fitness in your agent might really help answer those kinds of questions. So here are the 4 species of beetles that have been released since 2001 in the United States. And there have been some preliminary work done on potential hybridization of the beetles, mostly by Dan Bean. And what he showed is that Carinulata, which is the northern beetle found mostly in Colorado and northern latitudes, does not hybridize with the other 3 populations. However, there is evidence that the other 3 species can hybridize with only a moderate or slight reduction in F2 egg viability, depending on the exact species cross and also the maternal and paternal sex. So for the rest of the talk, we're talking just about these 3 species mainly. So this is a very busy graph, and I'm sure we've all seen distribution graphs throughout these last 2 days, but this one is really great because it shows the hybrid swarm that is occurring in Texas and also potentially in New Mexico, where you really have these 3 species coming together and mating. These data are from James Tracy, and they have been mostly identified morphologically, but we don't have any genetic data to back it up, and we have really no idea the extent of what's happening. So I guess I do want to say just quickly about this graph that we have collected populations all up and down these latitudes to really try and be able to quantify geographically the extent of the hybridization. So I'm doing these life history trait experiments where we have 9 crosses. So it's each of the 3 species that have been crossed with both male-female directionality, and I'll show you in the next slide more specifically if that sounds confusing. We are looking at a whole suite of life history traits. This is not even all of them. But basically, we're looking at traits related to fitness, so such as fecundity, oviposition timing, developmental time, mortality, and adult weight. And importantly, again, for biocontrol, we're going to be performing host choice testing with Aethel, which is a different species of tamarisk that, to my knowledge, until yesterday hadn't been attacked in the wild, but apparently they have started, so that'll be very interesting. But we're going to do host choice testing and also larval performance to development, all of these traits on the 2 different species of tamarisk. But unfortunately, like I said, this is going on right now. I have a technician in the lab who's covering for me so I can be here, so I only have results for 2 of these life history traits. So the scheme looks a bit like this. We have 1 species on the left, the parental, 1 species on the right, and then we have each of the crosses with the female from 1 species and the male from the other, and then the same thing vice versa. And we have this for all 3 species, so this is only 1 subset of all of the crosses that we have. We have them in buckets, so allowed to mate in a more group level setting. And then we made pairs so that we could follow families for the rest of the test. But in each case, we're always mating the same species together or the same hybrids together. And we now are down to the F3 generation where we're going to start measuring this larval performance on 2 different host plants tests. So again, the results are brief, but first I'd like to show you days until first oviposition. So this is a measure of fitness because it's how much time it takes an adult to begin ovipositing on the plant. And in this case, higher on the y-axis implies lower fitness because it's taking much longer. I would point out that here we have elongata and carinata. Elongata has just been doing really badly in the lab in general. We're not sure why. We don't know if it's inbreeding depression, if someone threw out a bunch of eggs. We don't know yet, but hopefully we can quantify it genetically and figure it out. But for now, we only wound up with 4 mating pairs, so the N is very low here, but we have at least 20 mating pairs for all of the others. So importantly, in this case, the crosses are doing well, but there does seem to be some evidence of directionality. When the female is coming from carinata, the fitness is actually lower than when the female comes from elongata in this case. This is sublineata and elongata. Again, this is the same results. Elongata is performing much worse. Here, the crosses are not different from each other, but they do have a lower fitness than the pure sublineata, so this goes with previous results. And here there was no difference between carinata and sublineata nor their hybrids. And then the second life history trait is total oviposition in the first 48 hours, so from the time they had their first oviposition until 48 hours after that. And in none of the crosses, there is no difference in any of the crosses here. So far, they're all performing pretty similar, but there is a lot of variation. So I know these are really preliminary results and we have a lot to look at, but one thing that I do want to say is that even though we see no consistent differences in these 2 life history traits between the hybrids and their offspring, it is not a null result. This can definitely be used. If hybridization, if there are no differences, then maybe it's not something we have to worry about. But it still needs to be something we take into account when we're looking at releasing biocontrol agents. And as I said, we still have a lot to look at, including host choice and developmental time. And we do still have the same predictions just based on previous work that we don't expect the hybrids to have as high a fitness as their parents. But that's why we're doing this, because we really don't know. And so now I'm going to pass it over to Amanda, who's going to discuss how we are genetically quantifying the levels of hybridization in the Southwest.
Okay, so you'll have to excuse me, I have to do this. Okay, so I show a picture of encyclopedias here because my point is to look at just one trait or just one gene is to read just a page of the encyclopedia, where a genomics perspective looks at the entire book, all of the collection of genetic information. And then we'll get into the biocontrol application, the Tamarisk leaf beetle, and how we can use genomics to understand hybridization and where we'll go from there. So what do I mean when I say genomic technique? Traditional methods that looked at genetic characteristics are allozymes, microsatellites, mitochondrial genes, and they are what were available at the time, but have a limited story to tell. So here we employ RADseq, restriction site associated DNA sequencing. Don't get caught up in that. The gist is that it's a reduced representation library. So you take the whole genome, and it's commonly known as next-generation sequencing or a genome scan. This enables us to catch genome-wide effects. So where mitochondrial genes are just maternally inherited, this captures more than that without the need for a reference genome. This is huge. And then it's cost-effective per locus. Since the Human Genome Project in 2008, costs have dramatically dropped, which allows us to apply these techniques to conservation, agriculture, and now biocontrol. And the result is that we get many, many more loci. So those loci come in two flavors, neutral and adaptive. When I am speaking of neutral, I mean those that inform demographic processes, population structure, migration rates, and admixture. So these are neutral, unrelated to selection, where adaptive traits are those related to selection. They tell us about selective sweeps, so how one gene can rise to fixation. Local adaptation, so we can see environment effects across a heterogeneous landscape. And then in the case of biocontrol, this is really relevant as a response to the novel environment. So from a genomic perspective, what is hybridization? Here I've represented the genetic material, the sum of all the genetic material as one line. But you can think of it as like the chromosomes. We can ask, and we are asking for this project, do hybrid genotypes outcompete parental genotypes? So we might not be able to see this in a cryptic species from the phenotypic level, but from a genetic perspective, you can pick up on loci from both species, and we could pick up potentially on hybrids replacing their parental genotypes, or just the opposite. Perhaps, as in this case, this is rainbow trout, where within an individual, as hybridization increases within this individual, or these individuals, fitness decreases. Alternatively, we can ask about the genetic composition, or here the genetic architecture of hybrid populations. You might imagine that all of the loci are mixed randomly and uniformly, but it might be the case that one parental genotype prevails and is admixed within an individual, or there are particular novel combinations that are more fit, and this could depend on the landscape or genetic effects within the chromosomes. It's, we get many more data for these questions. To look at the genetic architecture, we're developing species diagnostic markers. So this allows individual and population level of admixture. We can perform early detection of hybridization and identify those invasive alleles, where invasive alleles are just the parental alleles that have really high fitness. So in this figure, this is salmonid, and these are the chromosomes mapped out. So each chromosome in one individual and across the length of the chromosome. And these green lines are species diagnostic markers for one species. So this is what we're developing for the Tamarisk leaf beetle now across parental populations. In this first preliminary RAD-seq run, we are sampling 36 individuals. 6 samples are from within these parental species, illustrated by lab culture buckets. We also collected some samples from Greece for this particular study, and then 9 samples from within the hybrid zone. Really briefly, I'm going to go over the library preparation so you understand what I mean by that. We take our samples, extract raw DNA, and then restriction enzyme cuts throughout the genome semi-uniformly. So you end up with lots of fragments, and then barcode adapters are ligated. So this is an individual barcode which allows us to pool all 36 samples, and this is where the cost dramatically drops. You go from 36 wells to 1 Eppendorf tube. There's a shearing step so that we shear for fragments that can be sequenced on Illumina platforms and ligate the sequencing adapters. Our sequencing platform of choice is Illumina. We have a MySeq at the University of Idaho, and the result is tens to hundreds of thousands of loci across individuals. This analysis would not be possible without high-performance computers. I will not walk you through this entire figure. Suffice it to say, we align individuals so that can be genotypes, and we can pick out the single nucleotide polymorphisms. So the SNPs, or the individual differences across individuals. We can look at individual relationships and then population-wide relationships and get these diversity or heterozygosity scores. This structure plot is illustrating how we can tease apart. For example, this yellow is one pure population where green and red are showing admixture. And then we can also infer evolutionary relationships. So the library has been prepped, and I'm waiting for sequence data. This takes about a couple days. And in the future, this data set will allow us to tease out those species diagnostic markers, and we can start to build a null model to test hypotheses about selection. So we're working to characterize adaptation to latitudinal gradient because we suspect that there is evolution in life history. There's evidence to show that. There might be evolution in dispersal behavior. But we can tease out the differences between phenotypic differences and genetic differences. And then in the future, we can take a coevolutionary perspective where we can look at, I hope to look at, how Tamarisk and Diorhabda will interact over a long-term scale, and we can make long-term plans for Diorhabda and Tamarisk coexisting in North America. And this is relevant to climate change as well. Beyond Diorhabda, this is relevant to biocontrol systems in so many different ways. As Ellen alluded to, we should have a much better grasp on how ecotypes and hybrids interact in novel environments. How do we take genetic variation into account? This is a really important question, and the theory is still out. And then biological control systems also represent an excellent opportunity to study local adaptation and establishment of invaders. This work on biological control can also inform how we understand invasive species and manage them in the future. So with that, I'd like to acknowledge the primary funding for this project comes from the USDA NIFA grant that funds Ellen, and our advisors, Ruth Huffbauer and Paul Hohenlohe. This is us not finding any beetles in the beach and then finding them later in Greece. So with that, I'd like to take any questions.
It's well known that Tamarisk is hybridizing, and are you entertaining the possibility that hybridization of plant and its associated hybridization in the herbivores?
Associated? I'm not sure. I feel like geography is more related to why they're hybridizing, but I guess the only thing I would say to that is there is another group from South Africa that is doing my experiments, but sort of the opposite. They're taking all of the different Tamarisk hybrids and one species of beetle to see how they perform. But we do have some information from a PhD student who did who lists a sort of gradient, north-south gradient for the levels of hybridization. And I think at this point we could just do correlational work on that to see if there are an increase in hybrids and it's an increase in hybridization. I think though in the south, and someone can correct me if I'm wrong, it's mostly Ramosissima, and then there's more hybridization as you get further north. Is it the opposite way? Is it Chinensis? Okay, so there's more Chinensis in the south. So it's a little bit less hybridized in the area where we're seeing more hybridization.
I'd just like to add to that that it's possible that hybridization of beetles could lead to increased defoliation power on hybridized tamarisks, since the host specificity could allow them to defoliate across that range of hosts.
I have a question for Ellen. Are you all going to be looking at diapause adaptation as well as an egg hatch and things like that in hybrids?
Sorry, we need the same height. So I don't, for this exact study, I don't think that that's going to happen with the physical, like the phenotypic aspect of it, but diapause is definitely something we're looking at from the genetic level. So using the rad-seq techniques, we should be able, I don't think, a diapause is a suite of genes, it's not just one, but I think that this would allow us to look for signs of adaptation at lower latitudes, for example, in Crenulata, which would, because we are very interested in diapause necessarily, and that hopefully could shed some light on the genes responsible for that at lower latitudes. So not in the lab yet, but hopefully.
Not correlated to photo period.
Yeah. Yes. We are definitely going to get percent egg hatch on F2 and F3 hybrids, but as she said, not related to photo period. I have 9 crosses. There's only so much we can do.
Thank you.
Thank you.
Thank you.
All right, a couple quick reminders. Evaluations.